Hi, I'm Dennis Grant, and today we're discussing the voltage-gated or voltage-sensitive ion channel superfamily. We'll focus this discussion on reviewing how ion channels are studied to determine their physiological characteristics and how these are modulated by chemical agents. We'll also review the general structure of the voltage-gated ion channel superfamily, which includes sodium channels, calcium channels, and potassium channels. As well, using specific examples, we'll review examples of the biophysical properties of channels and how these characteristics relate to channel function and physiology. The biological membrane is a lipid bilayer which has a hydrophobic core. As a result, polar molecules such as ions have a reduced ability to cross the lipid bilayer membrane, and therefore require hydrophilic pores or channels that tunnel through the membrane. Generally, polar molecules can cross the cell membrane in one of three main processes: active transport, simple diffusion, and carrier-mediated transport. Active transport uses a transporter protein to pump molecules across the membrane. This process is used when a particle is moving against its concentration gradient from an area of low concentration to an area of high concentration. It's called active transport because the pump requires an energy source, such as ATP. Simple diffusion involves movement from an area of higher concentration to an area of lower concentration, and it doesn't require any added energy, only the random movement of molecules. However, polar, charged, or hydrophilic molecules can't diffuse across the membrane because the energy barrier of the hydrophobic core of the lipid bilayer is too large to overcome. Therefore, these molecules must rely on a carrier or a channel in order to cross, and this is called passive transport. Carrier proteins and channels rely on the property of molecules to diffuse down their concentration gradients, and therefore they don't require an input of energy. Ion channels differ from carrier proteins in a number of important ways. For example, they allow a much faster rate of transport, and they don't exhibit saturable kinetics. Ions are charged, and therefore are polar and unable to diffuse across the plasma membrane of cells, so they must rely on a carrier protein or a channel. Voltage-gated ion channels underpin the ability of excitable cells, like neurons in your brain, to communicate. Within a very small region, there can be tens to hundreds of channels, all with distinct properties of activation and modulation. Therefore, the ability to study channels in isolation and to determine their characteristics can be very challenging. Expression cloning is one of the techniques that's been used to isolate, identify, and study a gene, and subsequently its protein product. The combination of gene cloning, recombinant expression, and electrophysiology revolutionized the study of ion channels, excitable cells, and neuroscience in general. The combination of these techniques is used across the world in research and has resulted in multiple Nobel Prizes in medicine, physiology, and chemistry over the past 40 years. The basic idea of these techniques are highlighted in this slide, and we'll review them individually. Expression cloning has revolutionized cell biology, enabling the correlation of functional and physiological properties with specific gene products. In the ion channel field, it's been used to correlate channel function with specific structural families and subfamilies of ion channels. Briefly, RNA is isolated from a tissue that's known to express a protein of interest, and the messenger RNA is reverse transcribed into complementary DNA, or cDNA, using the enzyme reverse transcriptase. The cDNA is then inserted into a bacterial virus or bacteriophage, and bacteria are infected with the bacteriophage. The bacteria are then incubated on an agar plate that contains an appropriate growth environment for bacteria until they form a cloudy lawn but bacteria containing virus will soon die, producing a clear space or plaque on the lawn. The plate can be screened to find a plaque with a cDNA encoded by the gene of interest by producing a replica on special filter paper, followed by incubation with an oligonucleotide that binds to the cDNA, or with an antibody that binds to the cDNA's expressed protein product. Following sequencing, the cloned cDNA can be produced in large quantities by insertion into a bacterial plasmid and replication of the plasmid in bacterial cells to a high copy number, followed by isolation of the plasmid and its use in cell culture models to help delineate the gene product's structure, function, and modulation. Recombinant expression allows for the expression of the cDNA to be expressed in cells in study. The cDNA that was isolated by cloning can be co-expressed with a fluorescent marker called green fluorescent protein, or GFP. GFP is not normally expressed in cells, and it was originally isolated from the jellyfish. The cDNA to be studied, along with the cDNA for GFP, are transferred into cells that act as cellular shells for the protein of interest to be expressed and studied. The cDNA is provided to these cells at such a high quantity that the cDNA is overexpressed to a greater level than that of normal proteins that are usually expressed within the cells. The cellular shells are often cells isolated from tumor cell lines. These can be human embryonic kidney cells, otherwise known as HEK cells, Chinese hamster ovary cells, known as CHO cells, and even oocytes that are isolated from xenopus, which are frog eggs. A researcher can transfect one of these cell lines with the cDNA for the gene that they want to study and with GFP, and the cells will act as host and express the transcribed proteins. The GFP marker cDNA is included because the transfection of cells in culture can be inefficient. Therefore, the researcher can estimate the proportion of cells that are likely to express the protein for the cDNA of interest based on the percentage of cells that also fluoresce green under the microscope. Using techniques from electrophysiology, the cells can be examined for particular properties associated with the overexpressed protein of interest, which is then used to further understand the protein's function and modulation. If we were to overexpress the cDNA for a particular voltage-gated calcium channel, the data collected from the cells would be reflective of the overexpressed calcium channel protein. Electrophysiology is a technique that's used to examine the electrical properties of cells and tissues, and it's largely based on Ohm's law, since it measures changes in membrane voltage or potential and current movement across biological membranes as ion channels open and close, altering the movement of charged ions, which diffuse down their electrochemical gradients. An electrocardiogram is an example of electrophysiology at the level of the sum total of changes in ion movement via opening and closing of ion channels in the heart. Field potentials also measure the change in voltage from a population of cells. When studying specific channels of interest, researchers are more likely to use either patch clamp recordings or single channel recordings. Patch clamp recording includes recording changes in current flow or membrane potential as a result of many channels opening and closing within a cell. And single-channel recordings are the result of changes that occur from examining one specific channel in isolation. These electrophysiology techniques can be used to study channels expressed in our desired expression system, that is, those specific cDNA products overexpressed within our model cells, or it can be used to study native channel proteins in isolated neurons and cardiac cells from tissue. Furthermore, These techniques allow for the study of how these channels react or change their activity in the presence of drugs and chemicals. Therefore, they're also used to determine the pharmacological properties of these channels. The image on this slide shows a glass pipette attaching onto a single cell, creating a small pore in the membrane so that current and voltage can be manipulated and/or measured. The glass pipette is filled with dye, and you can see as it makes a hole in the membrane, it becomes one with the cell. The second image shows that the cells that are overexpressing our protein of interest can be exposed to various chemical agents, and corresponding changes in membrane potential and channel current movement can be measured. More specifically, the techniques of electrophysiology require a glass micropipette with an electrode that's filled with the appropriate solution. For example, it may be similar to intracellular fluid and contains ATP, glucose, and specific ions that are found in cells. The electrode is attached to a computer and an amplifier. The glass micropipette is sharpened to a very fine tip, which is then brought down and attached to the cell membrane. When the electrode attaches, a small pore is created, and a seal is made between the membrane and the micropipette, thereby making a circuit. This allows fluid to be exchanged, ions to flow, and current or changes in charge across the membrane to be measured. Again, this is based on the principle of V equals IR, where R, or resistance, is due to the membrane. If we're just measuring the overexpression of our protein of interest in a cellular model, there's a high probability that the changes observed in membrane voltage and channel activity is a result of modulation based on the specific experiment. For example, changing the voltage of the membrane results in channels expressed in the cell to open and close with a specific profile. However, if these techniques are used on cells that express multiple different types of channels, how would we be able to determine if the effects observed are due to one particular channel? The answer is that we can use known drugs and toxins to specifically block the activity of channels that are potentially still expressed in our cells, but that we're not interested in studying. We'll examine how we can do this later on when we talk about the role of tetrodotoxin, or TTX, in experiments. This toxin inhibits voltage-gated sodium channels. The left image shows the glass micropipette attached to the cell which is being studied. For single-channel recordings, the tip of the electrode is incredibly small, so small that it covers only one channel molecule. Changing the voltage applied to the cell results in measuring only the changes in current that result from this channel opening and closing. This is what's seen in the right image. Channels open very briefly. Also remember that opening of a channel is a stochastic or random event, so that when the channel is gated or activated, and in this case it's gated or activated by a change in voltage across the membrane, the probability of channel opening increases. And this, in fact, is measured by the electrode within the micropipette. The ramp at the top of the current trace indicates the change in the membrane voltage. In this case, the upward step indicates depolarization of the membrane. The last trace on this image shows the summation of openings from this channel over the time the membrane was depolarized. This summation of openings relates to the amount of ions or charge that moved through the channel during that period of time. This is called channel conductance, which is the measure of the amount of charge moving through a channel. Channels open briefly and quickly, and the activation of the channel will increase these brief openings. Wholesale channel recordings can also be completed with the patch clamp recording setup. The glass micropipette used for wholesale patch recordings is not as small as it is for single cell recordings, and through applying some negative pressure to the pipette, a small puncture or pore is made in the membrane, and the electrode inside the pipette can now measure the sum of current moving through all channels that are expressed in the cell, which open as a result of a particular manipulation, such as a change in the membrane potential or voltage. The current traces on the right demonstrate this. The ramp at the top shows how long and what voltage was applied to the cell membrane. That is, the membrane was depolarized. The deflection represents the channel opening and ions moving across the sum of all channels of a particular type in the cell. The use of these techniques, expression cloning, recombinant expression, and electrophysiological analysis of proteins expressed by specific genes from specific areas of the brain and heart, has moved our knowledge regarding voltage-gated channels forward, and has thus allowed for the study of how drugs and toxins can alter the function of these channels. So let's examine some of the major voltage-gated channels that underlie numerous functions within cells and are part of the voltage-gated ion channel superfamily. This superfamily includes ion channels that respond to changes in membrane voltage. These are the voltage-gated sodium, voltage-gated potassium, voltage-gated calcium, and voltage-gated chloride channels. However, there are also many channels that are like this superfamily, yet still distinct in many ways. This has resulted in the creation of a larger voltage-gated-like superfamily, which includes the non-voltage activated potassium channels and TRIP channels as examples. The general topology of voltage-gated channels is similar across the superfamily. That is, there are general similarities across the sodium, potassium, and calcium channels, with only minor differences that fine-tune function and modulation. We'll start our review of ion channels using the example of the voltage-gated calcium channel as the structural model. The alpha subunit is the main subunit of voltage-gated ion channels, and it's responsible for creating the channel within the membrane. The alpha subunit is quite large, and it can be modulated by the different ancillary subunits that interact with it. For example, ancillary subunits within the voltage-gated calcium channel include the alpha-2 delta, the beta, and, depending on the location of the channel, the gamma subunit. The alpha subunit itself is comprised of 4 domains that form a central region which can interact with the charged ion. Each subunit is comprised of 6 transmembrane regions with a loop called the P-loop between regions 5 and 6. The second diagram is a model of what the alpha subunit would look like looking down and into the cell. This slide shows the voltage-gated calcium channel looking transversely across the membrane. If we focus on the alpha subunit, each of the 4 domains is indicated by a Roman numeral from 1 to 4, and each is comprised of 6 transmembrane segments. In between segments 5 and 6 is a loop which traverses into the membrane. When all 4 P-loops come together, they help to form the central ion pore. This figure also begins to show the complexity of these channels, since they're modulated not only by membrane potential or voltage, but also by a variety of cellular modulators. The residues make up the structure of the voltage-gated channel, resulting in unique properties of the channel, and they allow it to undergo conformational shifts due to changes in membrane potential. These conformational shifts allow the pore or channel to briefly open and then inactivate, allowing ions to pass through and travel across the membrane for brief periods of time. Furthermore, the residues that comprise the P-loop or pore loop and the S5 and S6 regions determine selectivity for particular ions, therefore making the channels ion selective. Finally, there are minor modifications within the gene transcript which change the amino acid composition of the protein, creating channel subtypes that have slight variations in biophysical and modulatory properties. The combination of these biophysical properties results in a profile for each specific voltage-gated channel. The activation of the channel increases the probability of channel opening. Even though a specific membrane potential is the trigger for these channels, on their own they open randomly, and therefore the opening of these channels is considered to be stochastic or random. For example, the probability of a single potassium channel to be at rest when it's not activated is less than 10 to the minus ninth. And the membrane potential trigger or gating associated with activation and channel opening significantly increases the probability that more channels will be found open than closed. Using expression cloning, recombinant expression, and techniques from electrophysiology and molecular biology, researchers can examine the biophysical properties of channels and their putative functions in cells. Biophysics is an interdisciplinary science that utilizes the principles and methods of physics to study biological systems. As mentioned earlier, the structure, function, and properties of channels differ across families and also vary within families, creating channel subtypes. Between different biophysical profiles, anatomical locations, and expression, scientists can assign physiological relevance to a specific channel, structure, and function. The first biophysical property of a channel that we'll discuss is channel activation and opening. As indicated by the name of the superfamily, these channels activate, undergo conformational change, and open, allowing ions to cross the biological membrane down their electrochemical gradient as a result of a change in the cell's membrane voltage or potential. Remember that the majority of excitable cells have a resting membrane potential of approximately -70 to -90 millivolts. And at the resting membrane potential, there's an overall net balance of only a few channels activated and open, allowing for the maintenance of this membrane potential. However, as the membrane changes potential and the resting balance of ions inside and outside is no longer at equilibrium, specific channels expressed within the membrane become activated. These are the voltage-gated or voltage-sensitive channels. These channels are gated by or sensitive to changes in membrane voltage. The sensitivity of these channels to membrane potential or voltage is a result of the residues that traverse the membrane. This region of amino acids or residues that trigger the protein's conformational change as a consequence of changes in membrane potential is called the voltage sensor. The voltage sensor associated with the voltage-gated superfamily is highly conserved between species, as demonstrated by the alignment of amino acid sequence from the fourth segment of the third domain in sodium channels, calcium channels, and the Drosophila or fruit fly shaker potassium channel shown at the top of this slide. Each of the four domains of the alpha subunit is comprised of an S4 segment, which traverses the membrane and is comprised of a pattern of positively charged amino acids followed by two hydrophobic or nonpolar amino acids. As a result of these charged amino acids, the membrane undergoes a conformational change when the membrane potential changes or becomes more depolarized, that is, more positive. The positive charges of the S4 region play an important role during changes in membrane potential. And in the case of the voltage-gated sodium channel, this region triggers a conformational change in the protein as a result of charge repulsion. That is, as the membrane becomes more positive, the positively charged S4 regions shift due to charge repulsion of both the membrane and the S4 region being positively charged. Initially, the model developed to portray this potential interaction suggested that the S4 region moved up and down according to the membrane potential. However, more recent evidence and modeling suggests that the S4 region moves more like the backstroke of a canoe paddle, as seen in image B of this slide. As mentioned, the S4 region is similar but not identical across channel family and subtypes, but also due to other minor modifications in amino acid sequence elsewhere in the protein. What has evolved is that each particular channel subtype has a unique or different profile with regard to the exact membrane potential that results in channel activation. This minor difference begins to create a biophysical fingerprint associated with the channel subtype. This is demonstrated with the figure on the left side of this slide, and it corresponds to an electrophysiology experiment completed and shown on the right hand of the slide. Using whole cell recording of single channel subtypes, that is, only one channel subtype is overexpressed in an expression system, an IV curve can be created. This IV curve shows the relationship between the voltage applied to the cell's membrane and the ability of the channel to activate, open, and allow current to pass. In fact, the IV curve shown on the left-hand side stands for current voltage curve. The curve in the blue demonstrates the relationship for a subtype of calcium channel that results from the calcium channel alpha subunit gene called CAV1.2. The protein product associated with this gene is called the L-type calcium channel. What's important in this graph is that this current voltage relationship shows that as the membrane of the cell becomes more depolarized, that is, as it moves from -60 towards 0 millivolts, as shown on the x-axis, more of these channels become activated and undergo a conformational shift that allows more calcium ions to pass through the pore. And therefore, the current measured is larger, which is demonstrated on the y-axis. The figure on the right-hand side shows the voltage steps or membrane depolarization increase from -60 to +60 millivolts. The current trace gets progressively larger and then smaller as many channels open, and then fewer of the channels are able to activate at high voltages because the channels are now inactive. Peak current moving through this L-type calcium channel in this experimental protocol occurs at close to -10 to 0 millivolts of membrane depolarization. When this experimental electrophysiology protocol is repeated on cells expressing other specific receptor subtypes, like the CAV3.2 and 2.1 gene products, we see differences in the IV curve profile. Specifically, the calcium channel associated with CAV3.2 initially opens at more negative membrane potentials and peak current occurs at a more negative membrane potential, whereas the calcium channel subtype from the CAV2.1 gene opens and peaks at more depolarized, that is, more positively charged potentials. This begins the fingerprint of function and modulation that's associated with the various channel subtypes. The current voltage relationship shows us more than just when a channel begins to open and when peak current flow will occur. It also provides some information regarding channel inactivation and closing. This is also observed with the initial recording shown from the single channel recording technique. This type of data analysis was originally called noise analysis because of researchers examining the small peaks observed in the recordings due to single channels opening and closing very rapidly. By looking at the data on the right-hand side of the slide, it's understandable. Once again, using electrophysiology and an expression system, researchers were able to measure the voltage that increased the probability in channel opening. But they also interestingly observed that even in the presence of an appropriate depolarizing step, the channels did not stay open for the entire time the membrane was depolarized. The inability of the channels to open is the result of channel inactivation. Its observation is more pronounced in the summation of channel openings over the stimulated period of time in the final current trace. Similar to channel activation, channel inactivation also appears to occur as a result of a conformational change in the protein. The majority of what is understood about channel inactivation has been largely through modeling and examination of the voltage-gated sodium channel. Biophysical studies and models of these channels indicated that a peptide portion of the alpha subunit interacts at the intracellular pore region of the channel, that is the inside of the cell, and obstructs the channel opening. We can see this with the image on the bottom. At rest, the conformation of the S4 segments keep the channel mostly closed. However, with depolarization, the S4 segments shift, thereby opening the channel, which allows ions to diffuse across the membrane. After a few milliseconds, the inactivation polypeptide undergoes a conformational change and obstructs the channel to reduce ion flow. For the inactivation to be removed, another membrane voltage shift must occur via repolarization and/or a period of time must pass for the channel to return to its resting conformational state. For the voltage-gated sodium channel, this inactivation peptide is believed to be the C and/or N-terminal region of the protein. For some calcium channels, the inactivation peptide is believed to be the cytoplasmic loop between domains 2 and 3 of the protein. Many biophysical parameters have been determined using electrophysiology techniques combined with various expression systems. In summary, voltage-gated channels open in response to a change in membrane potential, with the majority of channels opening in response to membrane depolarization. And a specific threshold of depolarization must be met for channels to begin to activate and open. Channels will increase their probability of opening during this activation period, but over a period of time that's specific to the channel subtype, the channels will undergo additional conformational states that result in channel inactivation. At this point, even if the membrane depolarizing threshold is met, the channel will not reactivate and open. Overall, the voltage-gated channels result in 3 conformational states: open, closed but capable of opening, and inactivated, which is closed and unable to open. The name of each of the voltage-gated channels indicates that although there's a fair amount of homology within the superfamily, in fact each channel is selective for one ion species over another. In fact, when a channel becomes activated and opens, about 10 to the 6th to 10 to the 8th ions pass through per second. That's 1 to 100 million ions moving through the channel every second. So how is it that with all the ions on either side of the membrane, only one species of ion moves through an open channel? Well, in fact, this is not strictly the case. Ion channels are highly selective, with a voltage-gated potassium ion selectively allowing only 1 sodium for over 100 potassium ions. Thus, these channels are selective rather than specific. What makes them selective for one ion species over another? The selectivity filter. Within the pore region of the voltage-gated ion channel sequence, there's a series of amino acids that, based on size and charge, increases or decreases the probability of a particular ion species to move through the channel. This region of the pore is called the selectivity filter, and in the voltage-gated potassium channel, this region is comprised largely of tyrosine and glycine residues. Most of what we know about the selectivity filter has been through the work of Professor Rod McKinnon, who won the Nobel Prize in 2003 for his work on the structure of ion channels and the potassium channel more specifically. What Professor McKinnon's work demonstrated is that a channel's selectivity is based on the ability of ions which interact with water molecules in the aqueous environment of the body, and for these hydration shell bonds to be taken over by bonds between the ion and the residues in the pore region of the channel. For example, the sodium ion is approximately 95 angstroms, and when interacting through hydrogen bonds with water molecules, it's slightly smaller than 4 angstroms. Potassium, on the other hand, is larger and is greater than 1 angstrom on its own and larger than 4 angstroms with its hydration shell. In the case of the potassium channel, which has a pore region of about 1.4 angstroms, as the ion moves into the pore and the water molecules or hydration shell is shed, and these hydrogen bonds are now favorably replaced by bonds between the potassium ion and the residues of the pore, which helps potassium diffuse through the pore. However, sodium is too large with its hydration shell, and when these water molecules are removed, it's too small to interact with and form bonds with the residues of the potassium channel's pore region. As a result, the sodium is rejected from the pore region. As a result of the whole process, more potassium is able to move through the channel when it's open rather than sodium channels. As mentioned earlier, within each family of voltage-gated channels, there are channel subtypes. When we examine the current voltage relationships in each subtype, we can notice subtle differences about, firstly, which voltage channel subtypes begin to open, and secondly, when the maximal current conductance occurs. Since multiple genes encode different channel subtypes, the subtle biophysical differences between channels may be a result of the different genetic sequences that encode them. In the figure on this slide, we can observe the difference in channel activation and inactivation between two voltage-gated calcium channel subtypes. In this example, the CAV 1.2 channel opens slower yet remains open longer. That is, it's slow to inactivate. This profile is referred to as L-type or long-lasting channel current. In contrast, the CAV 2.3 channel opens rapidly and inactivates rapidly. The current from this channel subtype is referred to as R-type, as it's resistant to many of the common voltage-gated calcium channel modulators that were used to help characterize these channels. These biophysical distinctions are important because they underlie the distinct physiological roles of the different channel subtypes. Using whole-cell patch clamp recording of cells that express recombinant channels, we compared the biophysical properties of protein products encoded by 3 distinct genes. These channel subtypes result in 3 distinct profiles of opening and inactivation, and consequently in ion conductance, which is the amount of charge that moves through the channel when it's open. These distinct alpha subunit subtypes result in distinct channels that have distinct physiological roles and have distinct responses to pharmacological modification. This table summarizes examples of how different alpha subunit genes relate to different channel subtypes. Each subtype has a unique biophysical fingerprint and may also exhibit distinct expression patterns and physiological functions. Pharmacological agents can be used to selectively interact with specific channel subtypes, and this channel selectivity can increase clinical utility. An example of this is the L-type calcium channel, which can be created by 3 different genes. All L-type calcium channels have varying degrees of sensitivity to 1,4-dihydropyridines, but the most sensitive is the alpha-1C subtype, contains the CAV1.2 alpha subunit. This channel is also more highly expressed in vascular smooth muscle than in cardiac cells. Blocking the CAV1.2 alpha channel subtype with dihydropyridines can result in smooth muscle relaxation and reduces peripheral pressure. We see this clinically as a reduction in blood pressure. More importantly, because these more sensitive CAV1.2 channels are expressed in the periphery, dihydropyridines do not significantly affect heart alpha-1D L-type calcium channel currents, and therefore they don't slow cardiac contractility. This can be clinically important, especially in patients who already have a slow heart rate. So in summary, advancements in techniques in the late 1900s rapidly advanced the study of voltage-gated channels, their physiological role, and the development of selective pharmacological tools and agents that could modify these channels. Expression cloning used mRNA isolated from specific tissues to create cloned or copy DNA, which could be re-expressed in model systems. These recombinant expression systems allowed for overexpression of specific gene products to delineate specific voltage-gated ion channel characteristics. The combination of these techniques with those in electrophysiology resulted in correlating gene product structure and function with channel biophysical properties and physiological effects. These advances were mutually inclusive with the development and use of pharmacological tools, which were used to study and modulate the channels. The alpha subunit, which forms the channel and is modulated by co-expression of other subunits. The alpha subunit has 4 domains comprised of 6 segments each, a loop between segments 5 and 6 forming the pore, and the selectivity filter region, and the S4 region acts as the voltage sensor for activation. This conformational change results in channel opening, and it's matched with a second shift in the protein, which causes the channel to be obstructed for a period of time or until the protein returns to its closed state through membrane repolarization. An important aspect, both physiologically and pharmacologically, is that there are a variety of channel subtypes encoded by different genes. These different subtypes have distinct characteristics, locations, and functions. Moreover, an important area of pharmacology is in the development of pharmacological interventions that can differentially modify these channel subtypes so that they can be further studied in vivo and used for clinical applications. Thank you for joining me today. Now, with the basic understanding of how voltage-gated channels function, we can begin to examine how they can be modulated by pharmacological agents.